All right, so this is the final material before the midterm exam. And we're just covering eyes and ears. And you can see in this first part here, I kind of did some drawings. I used to try and teach this without drawing, and it was really just difficult for me. So these drawings, and I hope you can read my writing in here as well, because it'll give the information. But for eyes and ears here, we're starting with the eye, and this mentions that there are 3 layers of the eye. And the outermost layer is the sclera. This is the white of your eye. The middle layer of is the choroid and contains the blood vessels. And then the innermost layer is the retina. That's where your rods and your cones are, right? And all of those rods and cones, the little fibers that come off of them all join together and eventually go to the optic nerve, which exits out the back of the eyeball. The rods are used for non-color, low light vision. You do not see color with the rods because they do not detect color. The cones, you have red, blue, and green, of course, and so those are more daytime when you have more light because they require really more light to work well. All right, if you look at the front of the eye, this is kind of what we see. It just seems really simple from this point of view. But once again, I'm going to start this part right here. You can see the slung out. That's the sclera that we talked about earlier because that's the outermost layer of the eye. So that's the sclera. We have the iris, right? And this is pigmented, so it can be different color, different shades and so forth. And then the inside here that I've drawn black is actually an opening. And so this is where the light will actually pass through on its way into the eyeball. And of course, the iris here has smooth muscle, and this will control the diameter of the pupil. Finally, the sclera, all of this part out here, does not cover this part of the eye. This part of the eye actually has the covering on the outside is the cornea, which is a convex, clear surface that allows the light to pass through. All right, and this next image here, this is kind of a side view of the eye. So just to orient you just a little bit, if it looks a little bit odd, I've probably drawn this a little bit exaggerated, but that's okay. Eyeballs are not perfectly spherical anyway. So this is the front of the eyeball on this side, and this is the back of the eyeball on this side. And so if we remember up where we talked about the pupil, the iris, and the sclera, we can't really tell the sclera in this view because we didn't really draw it in. But the cornea is right here. We said covering that really front part of the eye. So that's the cornea. And as the light passes through, so this is light, it's going to pass through that cornea. We can't tell where the pupil is because I haven't drawn that either, but there would be a little hole here. The light would pass through it. And then here's the lens, right? So this is the lens here. And the lens can be elongated by pulling ligaments tight. Here's the ligament. So there's some smooth muscle out here that will contract. And I know this is 2 dimensions, but think if this is really 3-dimensional, then these ligaments actually go all the way around the lens. And so they can pull that lens and make it more thin, or they can relax and the lens will become a little bit more thicker. And that changes the angle at which the light passes through. And that's how we can focus the light then on the back of the eye. And we'll get to that in a moment. So the light passes through the lens. It actually bends just a little bit unless it's coming perfectly straight. And then it comes back and goes all the way back to the back of the eye. And you have at the back of your eyeball right here, this little tiny pit called the macula lutea. And you can see my definition here, area of the retina. So it's just a little tiny pit in the retina where we have a very high density of cones, which gives us highly detailed vision. Day vision though, right? Because remember, cones are for color and need a high level of light. But this is where whatever we're trying to focus on with our eyes, that's what we're doing, is trying to bring that light and put it right here. Any other light that passes through and then reflects and comes to the back of the eye is going around. This is all peripheral vision. The retina does surround the entire inside back here, but any of the rest of the light coming through is just peripheral vision. This little part back here, the macula lutea, is the central vision. Right, a couple more things I just want to point out before we move on from the drawing here, which I've colored all over now, is that there's this little jagged edge right here. That's because there's kind of this front chamber of the eye, and the light doesn't have to pass through this because this is actually going around and not in the middle. And so there's kind of the front part of the eye here, and then there's this whole back part of the eye. And this is filled with vitreous humor, which is really kind of a thick jelly that fills your eye. And it's important because your retina is out here, and the retina is not really attached except right here along this line, and then right back here at the back of the eye. That's the only places the retina is attached. So it's kind of like a blanket on a bed where you've only tucked it in a couple of places. The rest of it is loose. Well, then what holds that in place is this vitreous humor, which fills the back of the eye. And that's important because if that vitreous humor is dehydrated a little bit, or there's too much pressure, or there's different things, it can actually start to have an effect on the retina in some different ways. Finally, here's the back of the eye, optic nerve. So all of the rods and cones have little nerve things that will come back and join together, and they all come along this optic nerve, which eventually, of course, goes back into the brain, and then transmits information ultimately to the occipital lobe, where your vision center is located. So that was all a picture description. This just goes through it in words. So we have the pupil, where light passes through, the iris, which we said was pigmented, but there's smooth muscle that controls, that should say pupillary diameter. Let's fix that right now. Spell correct did not get that because papillary is actually also a word. The cornea, we said, is the covering on the surface of the eyeball, and that's where light passes through. It's only found on that front part of the eye. The lens is convex, so it's shaped like this. Okay. And it helps to focus the eye, or rather focus the light on the back of the eye. The light bends coming through the cornea, but we don't control the cornea. We do control the lens, and so that's how we're able to refract the light so it will focus at that macula lutea at the back of the eye. Right, we also mentioned that the lens, the thickness of it is controlled by some ligaments that are attached to smooth muscle around it. What we didn't mention, we did point out that the front chamber of the eye is really separate from the back chamber of the eye, and the back chamber of the eye is the part that had that vitreous humor. Front chamber of the eye is filled with a fluid, and it's called the aqueous humor. It's a much more runny fluid, and it creates something called intraocular pressure, or abbreviated IOP. And I presume that some of the students in their presentations may bring that up as part of their presentation because it's related to those disorders. This is also what they're measuring when you get that little puff of air at the optometrist or ophthalmologist when you go. They are measuring that IOP, and what they do is they want to track it over time and see if it changes, because that can be the result of something more serious or can indicate something. Well, why would there be a change in the intraocular pressure? Well, it's pretty simple. Production outpaces drainage. So you could have blocked drainage, or you could have overproduction, or you could have both. There's a number of different things that can happen, and I will leave it to the student instruction to go through that in more detail. All right, the next item here is I've mentioned the 3 layers of the eye. So we drew those up above. But here we go through them verbally. So the outermost layer, the sclera, it's the white of the eye, and it covers all of the eye except the front where we had the cornea. and then the back where the optic nerve is attaching. The rest of it is covered by this white. So the middle layer is the choroid, and then the innermost layer is the retina and its nervous tissue. It has the rods and the cones, and here again we have rods, black and white, low resolution, night vision, cones, red, green, and blue, high resolution, day vision, because you need more light actually to make them work effectively. We also already mentioned that the rods and the cones, they have nerve fibers that are associated with them, and that goes back and helps to form the optic nerve. We also pointed out this macula lutea at the back of the eye, where there's a very high density of cones, so that's where you're trying to focus the light. We also talked about the vitreous humor, filling the posterior, the back chamber of the eye, holding the retina in place. And now we get to start the ear. Okay. I apologize for my drawing. I always say my drawings are functional. They may not be accurate, but they are functional. They're certainly not to scale. And it may be really hard to tell that this is your ear on the outer part of your body. Here would be, you know, somebody's face would be drawn in right here. Here would be their eyes, right? Their nose, their mouth. And this is their ear sticking out off the side of their head. Just kind of to orient you to this figure a little bit. So this is kind of what we call the external ears, the part on the outside of the body, but there's a canal that then goes in all the way to where the eardrum or tympanic membrane is located. And this is known as the auditory canal. And attached to the tympanic membrane are the 3 auditory ossicles that, of course, now if you're transmitting sound, a sound wave comes in, makes the tympanic membrane vibrate, which makes the first bone vibrate, which makes the second bone vibrate, which makes the third bone vibrate. And then that sound wave gets transmitted over into the cochlear ducts. And these are actually found inside the temporal bone on the sides of your head. This whole area here is connected actually down into the back of the throat through the auditory tube. The name that most people are familiar with is still eustachian tube, but over years they've been trying to change the anatomy to kind of get rid of some of these names and make them more meaningful, if you will, to actually describe what they are. And there's a little tiny hole that opens into your back of your throat that extends up into this auditory tube. So remember, we were inside the cochlear duct, right? So inside those ducts, there are millions of these cells and some different membranes. And I'm actually leaving out some information here, but for our purposes, I think this is the important part. There's a part called the basilar membrane. So that's right down here at the bottom. There's another membrane that is sticking up right here, and that's called the tectorial membrane, and it's a really stiff membrane, whereas the basilar membrane is going to vibrate. All right, now on the inside here, sitting on the basilar membrane is an inner hair cell, and there are these cilia that protrude from this inner hair cell, and they're kind of a different length here. And attached to that inner hair cell is an auditory nerve fiber. All right, so that's kind of the basic anatomy. Now, we mentioned that a sound wave is going to come down the cochlear duct. And when it does, and again, we're missing some steps, so that sound wave is going to cause this basilar membrane to vibrate. It's not going to make the tectorial membrane vibrate because this tectorial membrane is really stiff. The basilar membrane starts to vibrate, and it starts to vibrate, it starts to push this hair cell and makes it go up and down, pushing these cilia up into the tectorial membrane. What then happens is these cilia will bend, and that causes a signal. That's the signal, which is then transmitted along this auditory fiber. Now, different places within the cochlea, you sense deeper sounds, mid-pitch sounds, higher sounds, and that's actually how we can tell sound pitch is different hair cells will get activated in different parts. And then the loudness of the sound is how many of these hair cells get activated, or are there higher threshold hair cells that don't even fire until there's a really loud sound. And so we can interpret the volume of the sound as well that way. So if you just look right over here, this is kind of the summary of what I just explained about how that sound signal is generated. So again, in verbal terms, we have the ear anatomy. We have that auditory canal, which directs the sound towards the middle ear. The tympanic membrane vibrates in response to those sound waves and passes that signal onto the auditory ossicles. These are actually found in the middle ear. The tympanic membrane is kind of the dividing line between the external ear and the middle ear. Those auditory ossicles then vibrate and pass the signal to the cochlear ducts. And those cochlear ducts are fluid-filled tubes. And we didn't really get to draw that before. But of course, fluid will also pass a wave along. And so that's what happens. It vibrates, there's a wave that causes that basilar membrane to vibrate. And that causes those hair cells to move up into the tectorial membrane. And they will bend, and you're going to sense sound as a result of that. Right, I've been through these details just now, but just to remind you that it's those, that they're called the inner hair cells because those cilia are protruding from them, and they push into that tectorial membrane. And bending them, that's a mechanical signal, really responding, if you will, to a mechanical stimulus, which was the sound wave coming into the ear. And that's all pretty basic, but I think that is enough, at least to describe the basic anatomy and physiology of it, to better understand some of the different disorders that can happen with aging.